Let's continue our discussion on Genome Enhancer. If you haven't watched part 1 or other videos in this series, please make sure you check it out. I will be adding the link to the part 1 and other videos in the series in the description section below. So once it finishes running, you should be seeing something like this where you see the status complete and you get an option to view a brief report or you get the complete comprehensive report. So I'm just going to click on the full story here and it opens up this report which is in the format of a research article which has its abstract, introduction, data, results, and describing each section of the detail of the results in further detail along with the figures and plots, as well as describing the methods that are used. And lastly, they also provide you with the conclusion. So we are going to go over the details of this report as there's a lot of information that is contained in this report. So we are going to go over each of the section and briefly discuss various insights that have been reported in this final output report. So starting with the abstract, in the abstract, it mentions the study, the context of the study, and describes the goal of the pipeline, and also briefly describes the steps that it performed to identify the interesting or the potential markers. So for our analysis, it identified transcription factors, and these are the transcription factors that it identified among the which would be involved in regulation of the differentially expressed genes between RNX knockout and the RNX wild type lines. And these are the most promising candidates. And performed the network analysis and identified the most promising molecular targets, as well as these targets could further be of importance to for further research or drug development. And it also identified based on these targets the most promising treatment candidates, which could be further tested. So this was just the overall gist of the insights and the finding that it generated through this pipeline. So followed by the abstract, we have introduction, where they more comprehensively describe the upstream analysis approach and how it identifies the transcription factors, thereby identifying the master regulators, and also helping us perform network analysis, identify more targets which can be targeted using certain drug candidates. Followed by the introduction, we have the data section. So typically, this is the data that we provided to the Genome Enhancer pipeline. We provided the transcriptomics and epigenomics data, and basically it just shows how we have our experiment set up. So basically how we divided our samples into wild type and knockout. And then the Genome Enhancer performed differential expression analysis on them, and it moved ahead to finding the transcription factors, master regulators, performing network analysis, and so on. Followed by the data, we have the results section, and in this section we have a lot of different types of results that have been reported. The first section here is identification of target genes. So the first step this pipeline performs is to identify differentially expressed genes between the two set of samples or multiple conditions that you're comparing. In our case, we identify genes that are differentially expressed between RNX1 knockout and RNX1 wild type MCF10A cells. In order to identify differentially expressed genes, they use lemma. So basically here they provide the description of the tool and all the parameters that they've used to identify the differentially expressed genes. So they identified around 3,602 upregulated genes with the log fold change greater than 0.1, and out of which 2,032 genes were found to be significantly upregulated, and this was the p-value threshold that they used to identify that. These were the down-regulated genes that they identified with this log fold change threshold, out of which 2,339 genes were found to be significantly down-regulated genes. Followed by that, they have given us the tabular compilation of the upregulated and downregulated genes along with their metrics. You can click on the See Full table to get access to the complete list of the upregulated and downregulated genes, and these tables can be exported using this button here. And there are various formats that this can be exported to your own computer, so you can save this data for further or later use. And these tables are also linked in the supplementary section. So as I said previously, this report is very much in the format of a research article, with all the data, figures, and insights and findings, along with the conclusion reported in one document. After having identified differentially expressed genes, the pipeline next tries to map them to the ontology terms like gene ontology, disease ontology, and ontology terms associated with signal transduction and metabolic pathways. This is to give us a sense of the enrichment of these terms in our gene sets and basically giving us an idea on what these genes are associated with in terms of biological processes, diseases, or and what signal transduction pathways and metabolic pathways these genes might be involved in. So the first thing that they provide us is with a heat map of these differentially expressed genes, showing the expression in a form of a heat map between our wild type and the RUNX1 knockout samples. So followed by that, in a form of a tree map, this visualization shows us the enrichment of the ontology terms, the gene ontology terms that are associated with the biological processes. So basically, we get to see what biological processes are enriched, or rather what gene ontology terms associated with a certain biological processes are enriched in our upregulated genes. Followed by that, they do something similar with the pathways. So this pie chart basically shows us the composition of the gene ontology terms associated with a particular pathway. So this can be helpful in visualizing what are the enriched terms that are associated with a particular pathway, which are associated, which could involve our upregulated genes. Followed by that, we are also able to see the ontology terms associated with the diseases, and we get to see the distribution of these ontology terms in our upregulated genes. Followed by that, they have repeated the same visualization process and the same process for the down-regulated genes as well. So they show us the ontology terms enriched with associated with biological pathways, followed by what transpath pathways these terms are enriched in. And also followed by that showing us the diseases. So one of the observation in this case is that a lot of ontology terms associated with breast neoplasms are seem to be enriched in our downregulated associated with the downregulated genes, and this seems to be a more promising or adding further confidence in this analysis that it's able to identify these ontology terms, because the targets of the genes that we identified are comparing a target that plays a crucial role in breast neoplasms or breast cancer. And finally, they basically have this plot where it summarizes the overall results of gene ontology and identifies the most significant functional categories which are overrepresented amongst the observed differentially expressed genes. There are several ontology databases that have been used, and if you require a detailed description of these databases and what information it holds, you will find a link in the description section which describes them in further detail. After performing the function analysis, the next step is to perform transcription factor binding site analysis. And this is performed in the regulatory regions of the target genes by using the transcription factor binding motif library called the transfac. They also search for composite modules, which essentially act as condition-specific enhancers for target genes in their upstream regulatory regions. And they also identify transcription factors which are regulating the activity of these target genes through such enhancers. So the idea here is that this step attempts to identify the enhancer region of the target genes, and these enhancer regions act as targets for multiple transcription factors which bind in a cooperative manner to regulate the genes of interest. As we have seen previously, there is usually several transcription factor coordinating and acting in a cooperative manner to regulate the genes. So the algorithm identifies the transcription factor that binds to these usually clustered regulatory regions and cooperates to provide a synergistic effect in influencing the gene's regulatory process. So the pipeline was able to identify two composite modules in the upregulated genes. So basically, in order to identify the composite modules, the pipeline chose the top 300 most significantly upregulated genes, and it provided it as an input to the CMA algorithm. So here we have the details of both these modules. So in the blue boxes are the position weight matrices that corresponds to the transcription factor, and these transcription factors were found to be cooperating in and acting as a composite module, which basically acts as an enhancer and regulates the activity of the target genes. Um, if you're not sure of what the position weight matrices are, then I will recommend you to watch the first part of this series of videos, and you will be finding all the videos and the links to them in the description section below. In the first part of this series, I have covered the basics of the transcription factor binding sites, what are motifs, what are position weight matrices. So if you're not sure what those are, then I will recommend you to check that out. Followed by the modules, there are various metrics and the scores available from the CMA algorithm for these modules. And here we have a histogram which basically separates the yes set and the no set. So basically this histogram shows how the identified modules separate the genes from the yes set and the genes from the no set. Followed by that, we have a tabular compilation of the top 10 upregulated genes with identified enhancers in the regulatory regions along with the CMA score. The CMA score is essentially the score from the CMA model of the enhancer identified in the regulatory region. And along with that, we have been given factor names which are associated with the enhancer regions for each of those genes. If you're interested in seeing the full table, you can click on the see full table, and again you get the entire table that you can explore if there is any specific genes that you want to look at. And this table can be exported to your local system in various file formats. Similarly, the composite modules are identified among the top most significantly downregulated genes, most top 300 most significantly downregulated genes. And similarly, we get another histogram which is separating the yes and the no gene set. Followed by that, we have been provided with a similar tabulation of the CMA scores and the factors associated with the identified enhancers amongst the downregulated genes. On the basis of these enhancer models, the pipeline identified transcription factors that potentially regulate the target genes of interest. They found 20 transcription factors controlling expression of upregulated genes and 13 transcription factors controlling expression of downregulated genes. Here they have provided compilation of each of those set of factors, and they've also provided some additional metrics like regulatory score and yes and no ratio. The regulatory score is the measure of involvement of a particular transcription factor in controlling the expression of genes that encode master regulators. And the yes and no ratio essentially describes the level of enrichment of binding sites of a particular transcription factor in the regulatory target region. They provided this diagram which basically represents the key transcription factors which are predicted by the pipeline to be potentially regulating differentially expressed genes in our study. So it was identified that several of the SMAD family transcription factors, SP1, were among the few that are the key transcription factors regulating the expression of our identified differentially expressed genes. So after having identified key transcription factors, next the pipeline identifies master regulators. Essentially, these are the molecules that regulates the transcription factors that are identified, and these serve as key candidates for therapeutic targets. So in the first table, the pipeline tabulates the master regulators that govern the regulation of upregulated genes in RNA-Seq knockout cells. In addition to different kinds of information like the gene symbol and the gene description, they've also provided certain metrics like log fold changes and the total rank. How the total rank is calculated is provided in the description above the table, but basically the way you interpret it as is the smaller the rank, better is that respective master regulator. Similarly, scrolling further down, here they've provided the list of master regulators that may be regulating the downregulated genes in the RUNX1 knockout cells. Followed by that, they have provided a comprehensive intracellular regulatory pathway map, which basically displays the connections between identified transcription factors, which may play roles in regulation of differentially expressed genes, and selected master regulators, which are responsible for regulating these transcription factors. So I'm going to click on this map and make it a little bit bigger so we will be able to visualize the map. So this is essentially the intracellular pathway map showing the regulation in the upregulated genes. So here the red boxes indicate the master regulators. The blue boxes indicate the transcription factors, and the orange and the blue highlights are the molecules basically that are encoded by the upregulated genes and the downregulated genes. The orange highlight is for the upregulated genes, molecules encoded by the upregulated genes, and the blue highlight is for the molecules that are encoded by the downregulated genes. So this comprehensive map gives you an overall idea on how your master regulators are interacting with your transcription factors and with various other molecules, and what roles and what pathways are involved in regulating certain genes. So similarly, we also have a similar map drawn out for the regulation of the downregulated genes, and various transcription factors and master regulators in this case seem to be interacting in this complex network, regulating the expression of some of the crucial genes that may be playing a role in breast neoplasms. After having identified master regulators, next the pipeline tries to quantify the potential for these master regulators to be drug targets, as well as trying to predict the molecular mechanisms of the studied pathology. So in this section, basically they provide us with the prospective drug targets selected from the full list of identified master regulators, which are filtered by something called as druggability score. Now, the druggability score is calculated two ways using the human PSD database and the PASS software. The details of these databases and software can be found in much detail in the documentation. So if you want to learn more about them, I recommend you to refer to the documentation. In addition to that, they also provide metrics like log fold changes and the total rank. Again, the total rank, the smaller is the rank, the higher or a better suitable a particular target is for being a drug target. Scrolling further down, here they provided a schematic representation of the main molecular mechanism that is being predicted based on the master regulators that they identified. So here the solid lines indicate the intracellular interactions, and the dotted line indicates a positive feedback loop. And here you see many components interacting with various other molecules and which transcription factor it affects and what are the processes that those transcription factor are they end up affecting. So this provides you with a great starting point and generates a lot of insight into what could be going on, and would be able to help you narrow down to focus on certain key master regulators or certain transcription factors that you would want to investigate further. In the last section of this report, the pipeline identifies potential drug that may be a suitable candidate to target the identified regulators, master regulators, or transcription factors in the previous steps. And these potential drugs could be from four categories: FDA approved drugs, or these drugs being currently used in clinical trials of the studied pathology, or repurposing drugs in clinical trials for other pathologies. Drugs could be predicted by PASS algorithm, which are identified to be active against the identified targets, as well as in the particular studied pathology. In our case, it would be breast neoplasms. And in the last category would be the drugs that are predicted by this algorithm to be active against the identified drug targets, but in other categories. So basically, you're getting suggestions and identifications from the pipelines regarding a lot of types of active components that could be investigated further, and thereby a therapy could be developed based on the targets that are identified. Followed by that, we have supplementary drug information. We also have the method, the conclusion, and followed by the conclusion, we have the methods, which is a description of all the databases and the methods that are used in this pipeline. And finally, we have the references as well as the supplementary material, which are the links to all the various tables that are mentioned in our report. In conclusion, using Genome Enhancer, we were able to process multi-omics datasets and identify the crucial role of RUNX1 in breast neoplasms. The pipeline pre-processed and statistically analyzed the data and performed differential gene expression analysis. In addition, function analysis and enrichment analysis was also performed on the identified differentially expressed genes. Further, the pipeline identified master regulators, composite modules, and provided a detailed signal transduction network. In the network, top master regulators were highlighted that played a crucial role in the molecular mechanism of the studied pathology, and a proposed schematic representation of the main molecular mechanism was also provided. Finally, druggability potential of the identified master regulators was evaluated and potential drugs were identified. The pipeline provides the most promising molecular targets for further drug repurposing and drug development initiatives. So that brings me to the end of this video. As I said previously, if you haven't watched the previous videos in this series, you will find the link to those in the description section below. If you enjoyed the video, please consider giving it a thumbs up. Do not forget to subscribe to my channel for more bioinformatics related content. If you want to find out more details about Genome Enhancer pipeline and explore its other applications for the analysis of various combinations of input omics data in different pathologies, you are welcome to explore geneXplain's YouTube channel and the Genome Enhancer playlist in particular, which I will be linking in the description section below. If you have a question or a topic you would like me to cover, please leave it in the comment section below. Share this video with your friends and coworkers if you found it helpful. I appreciate your support and I'll see you in the next video.